Hi, my name is Brian Jaros, and I will be a senior at Avon High School. During the summer, I participated in the Medical Laboratory Internship Program, working in the histology lab at the Cleveland Clinic main campus. The title of my research project is The Utilization of Diffquick in the Identification of Ganglion Cells at Frozen Section. Hirschsprung's disease is an irregularity of the gastrointestinal tract, specifically in the large intestine. This abnormality results from a lack of nerve cells, known as ganglion cells, in portions of a patient's intestine. Hirschsprung's disease affects about 1 in every 5,000 births and may range in severity. The deficiency of ganglion cells in the intestines of Hirschsprung's patients leads to limited bowel movements of stool. Patients, who are almost always newborn babies, will likely suffer from severe constipation and intestinal obstructions within 48 hours of birth. A swift treatment of the disease is required, as prolonged intestinal blockage leads to infection and, in most cases, death. In a healthy newborn, ganglion cells have grown throughout the entire large intestine of the fetus, beginning with the colon and ending at the anus. In babies with Hirschsprung's disease, ganglion cells have ceased growth in certain portions of the intestines. The amount of nerve cells that have not grown varies from patient to patient. It is not known why the growth of these nerve cells stops. The picture on the left represents a healthy large intestine where ganglion cells are present throughout. The picture in the middle demonstrates a mild case of Hirschsprung's where ganglion cells are missing from a small portion of the intestine. The picture on the right exhibits a severe case of Hirschsprung's disease where nerve cells are absent from a majority of the intestine. The most common and effective method of diagnosis for Hirschsprung's disease is a rectal biopsy, where a tiny piece of intestine is removed from the patient. The tissue is then processed, stained, and reviewed under a light microscope by a pathologist. In order to form a diagnosis, the pathologist observes the tissue for the presence or absence of ganglion cells. This identification must be performed rapidly in order to confirm a diagnosis and alleviate symptoms of the disease. The identification of ganglion cells is currently hindered by the routine H&E stain used on the intestinal tissue. H&E, or hematoxylin and eosin, stain ganglion cells the same bright pink as surrounding smooth muscle and nerve fibers. The ganglion cells become undistinguishable by color, delaying the identification process. In addition, ganglion cells are small and immature in infants and appear similar to endothelial cells. The study was performed in order to compare the routine H&E staining to the Diffquick staining technique in the identification of ganglion cells. The Diffquick stain was evaluated as a complement to the H&E stain on frozen sections of tissue. The photo at the top of the slide represents tissue stained with H&E. The tissue stains a light pink color, while the Diffquick stains a tissue a more purple color, as seen in the bottom photo. The first step of the study was to submit 11 samples of ganglionic colon for frozen section. The tissue was cut into routine 5-micron sections using a cryostat machine. 2 levels of each sample were cut to be stained with H&E and Diffquick. The slides to be stained using Diffquick were air-dried prior to staining. The tissue was first placed in Diffquick fixative, chemically known as methanol. After 30 seconds, the slides were transferred to Diffquick solution 2, where they soaked for 30 additional seconds. The slides were rinsed with tap water to remove any excess Diffquick and were placed in absolute alcohol to rapidly dehydrate the tissue. Finally, the slides were cover slipped. The slides to be stained using H&E were briefly fixed in 95% alcohol and rinsed with water. The tissue was stained in hematoxylin for 1 minute. After staining, the slides were first rinsed in tap water to remove excess hematoxylin and then in ammonia water to add a blue tint to the tissue. Following the ammonia water, the slides were placed in tap water and 95% alcohol for 10 seconds each. The tissue was stained in eosin for 10 seconds and then briefly rinsed twice in 95% alcohol and twice in 100% alcohol. Finally, the tissue was soaked in 2 changes of xylene until the slides appeared clear and were cover slipped. The tissue samples were submitted to be reviewed by Dr. Bennett and Dr. Gonzalo. 14 total samples were submitted, however, only 11 of these samples were used due to deficient staining in 3 of the sections. The reviewers were asked to count and record the number of ganglion cells they could find in each stain. The number of ganglion cells found in each type of stain was totaled for each individual reviewer. Reviewer 1 counted a total of 135 ganglion cells in the H&E stained tissue and 194 ganglion cells in the Diff-Quick stained tissue. Reviewer 2 counted a total of 104 ganglion cells in the H&E stained tissue and 146 ganglion cells in the Diff-Quick stained tissue. In both cases, more ganglion cells were identified in the Diff-Quick stain than in the H&E stain. The average mean of ganglion cells counted in each stain was calculated using the previous data. The average means of reviewer 1 and 2 in the H&E stains were 12.3 and 9.5 respectively. The average means of the Diff-Quick stains were 17.6 for reviewer 1 and 13.3 for reviewer 2. The mean number of ganglion cells in the H&E stain by both reviewers was 10.9 and 15.5 with the Diff-Quick stain. Here we can see the difference between the two types of stains studied. The picture on the left shows the H&E stain of intestinal tissue. The cytoplasm of the ganglion cells is stained pale pink and the nucleus is purple. The color coordination is similar to that of the surrounding pink muscle and nerve fibers and the purple endothelial cells, slowing the identification of ganglion cells in this stain. The Diff-Quick, shown on the right, stains the cytoplasm of ganglion cells bright blue and the nucleus deep indigo. The contrast between the bright blue cytoplasm of the ganglion cells and deep indigo nuclei with prominent nucleoli facilitates the identification of ganglion cells against the surrounding tissue. The Diff-Quick stain could provide a useful tool in the identification of ganglion cells. Adding a Diff-Quick stain to the routine H&E stain at the frozen section would further enhance a pathologist's ability to identify ganglion cells. In addition to enhancing the identification of ganglion cells, the Diff-Quick stain is relatively cheap and easy to perform. As an anecdotal observation, 4-micron thick sections of tissue are more amenable to Diff-Quick staining. I would like to thank and acknowledge Dr. Bennett, Dr. Gonzalo, and Lynn McDonald for their help in my research. I would also like to thank Nedra Starling and the Office of Civic Education Initiatives for giving me the opportunity to perform this research. Thank you.